Network Symposium at the Creek. My name is Inês Sequeira. I'm a group leader at Queen Mary University and a co-founder of the network.
I'm another co-founder of the network. My name is Davide Danovi, and we're here to interview Michele de Luca, who was a keynote speaker at the event. Michele, thank you for being with us. What do we learn from the interaction between clinicians and academics? And in particular, what is the right point between making sure that the regulation are set, but at the same time innovation is not stifled?
Well, actually, these are two different questions. Okay, so the first question is fundamental. If we talk about the new generation of therapies, which are not drugs, classical drugs, there is no surgery, is advanced therapies. So it's biological drugs that are made with the cells, different type of cells, okay? No matter whether you do cell therapy or gene therapy or tissue engineering. So in order to do this, you have to know the developmental biology of the tissue, you have to know the physiology of that particular stem cells, especially for renewing tissue, and you have to control those stem cells if you want to do properly cell therapy and gene therapy. Then you have to go into the clinics, okay? And the patients usually will tell you whether you're doing the things properly, and will teach you a lot of things also about the biology of the system you are using. Well, that's the first question. The second question is a bit more complicated because, you know, it stems from the definition of these advanced therapy as drugs, and therefore they have to be treated as usual drugs. But many of these advanced therapies, with stem cells particularly, not to say about gene therapy using those stem cells, are for rare disease, very rare disease. So it's a personalized type of medicine, which is not comparable to a pill, to classical drugs that you distribute in all the pharmacies. So am I saying that we don't need the rules? Not at all. We need the rules. Only those rules should be tailored, should be on purpose tailored for the type of advanced therapy you do, in order to facilitate within a proper control, but facilitate the development of this therapy, because otherwise they are very difficult to develop.
One of your work on using genetic and cellular therapy for epidermolysis bullosa was one of the breakthroughs in the stem cell field. And today you mentioned that it was also the most emotional moment for you as a scientist. Can you tell me more about this work, this breakthrough, and how you felt as a scientist at that moment?
I didn't expect this question. Yeah, it is true, because even though, as I said, even though we were used to this because of the burn patients, the ocular surface, but the story of this kid was particular, was escaping from the war, was dying, and on the top of this, I felt a lot of responsibility, because I was the one that said yes to these people to this colleague in Germany to try this, but I knew it was difficult, okay? So is a number of reasons that you put them all together, which I said when we saw that the skin was developing underneath those gauze and the kid was recovering all of a sudden, that was absolutely amazing for me, and that's probably. Let me put it this way, I tell you with one sentence, that kid for me it means my entire scientific career. For me it's enough, that kid, to justify all the adults I've been going through the development of these gene therapy trials.
And so what would you say to the scientists who start now? What is your message to our London Stem Cell Network fellows?
For the future stem cell biologists.
Well, actually, I have a basic message that no matter whether you want to do pure research or translational medicine, don't forget the biology of the cells you're dealing with. The basic research and the knowledge, deep knowledge of the biology of the cells you are knowing is instrumental in order to have any clinical result, okay? And is important, is crucial. Many times in the world right now, people are using stem cell, so-called, or pseudo, as I call it, pseudo stem cell, improperly. You have to start from the developmental biology to the stem cell biology, and it is important. Having said that, that would be already sufficient. My personal experience is that if you can then translate those knowledge into a therapy that can help people with no therapy, that is one of the most spectacular things that can happen to you.
And thinking about not just cellular therapy, but all the repurposing of drugs that are used for other diseases, in the case particularly of epidermolysis bullosa, is there something that can be used to be combined with cell therapy, or drugs that are used, for example, in psoriasis that could help these
you mean the other therapy that are proper? Well, you see, epidermolysis bullosa is a genetic disease, so is monogenic, so we know exactly the gene that is involved and all the mutations. There are a lot of treatments that are starting from the medication. There are spectacular medications that are being developed by many companies that are used to the industrialized country. Actually, we receive sometimes kids that comes from underdeveloped countries, and the way they do the medication is absolutely unacceptable. But they don't have the access to these therapies, which is crazy, because they are kids. Both are kids. The one that lives in the underdeveloped country and the kid that lives, in Rome or Paris or London. Then, since the skin is so complicated, for instance, in the epidermolysis bullosa, we have the lesion in the fingers and everything. So probably you can combine different type of therapies to better help this. We are devoting our research to the gene therapy because, being a genetic disease, correcting the stem cells is the only way, there is no other way to fix the skin permanently in one clinical application. It is the most difficult things to do. But certainly, during this process, you can combine different therapies.
Thank you very much for being with us.
Was it okay?
That's great. Thanks.
Perfect.
Thanks.